we stand at the precipice of an era defined by our newfound ability to rewrite the very blueprint of life. For millennia, human evolution has been a slow, undirected dance orchestrated by natural selection and random mutation. Diseases ravaged populations, traits emerged by chance, and the genetic lottery determined much of our individual and collective destiny. But that ancient rhythm is now being profoundly disrupted by a revolution born in the laboratories of modern science. The age of advanced genetic engineering, no longer confined to the theoretical realm of science fiction. The power to precisely modify the human genome has moved from improbable fantasy to imminent reality. At the heart of this transformative shift lies CRISPR-Cas9, a gene editing tool so revolutionary, so precise, and so astonishingly accessible that it has democratized the ability to edit DNA in ways once unimaginable. This technological leap represents not just an incremental improvement, but a paradigm shift, granting humanity an unprecedented degree of control over the biological heritage of our species. The implications of this power are breathtakingly vast and complex. On one hand, it holds the promise of eradicating inherited diseases that have plagued humanity for generations. Conditions like cystic fibrosis, Huntington's disease, and sickle cell anemia could potentially be corrected at their genetic root, offering cures rather than just treatments. Imagine a world free from the suffering caused by these relentless genetic scourges. A world where the future health of children is not dictated by the cruel hand of chance. This therapeutic potential alone marks a monumental step forward for medicine and human well-being. Yet, as with all powerful technologies, the capacity for profound good is inextricably linked with the potential for profound ethical peril. As our scientific prowess expands, so too does the scope of our moral responsibility. The line between therapy and enhancement blurs, raising deeply unsettling questions that demand our immediate and thoughtful consideration. This essay will delve into the future of genetic engineering, navigating the exhilarating possibilities and the chilling implications, forcing us to confront the most profound ethical minefields of our time. Central to this discourse are two interconnected, highly controversial concepts: designer babies and widespread human enhancement. The term designer baby conjures images of parents selecting traits like eye color or athletic prowess from a genetic menu. While initially focused on preventing severe genetic diseases, the technology's precision and potential scalability inevitably lead us down a path where selection for non-medical traits becomes increasingly feasible. What begins as the noble pursuit of health quickly edges towards the desire for perfection, or at least optimization. Beyond the creation of genetically tailored offspring, we must also grapple with the broader concept of human enhancement. If we can correct a disease, can we then boost intelligence, prolong lifespan, or even augment physical capabilities beyond natural human limits? The very definition of human itself comes into question when we possess the tools to fundamentally alter our genetic makeup. This isn't merely about treating illness. It's about potentially redesigning what it means to be human, with all the inherent dignity, frailty, and diversity that currently defines us. This profound power unleashes a torrent of ethical dilemmas. Where do we draw the line? Is it ethically permissible to edit genes to prevent a fatal disease? What about a predisposition to a serious condition? What about a non-life-threatening genetic trait, such as deafness or dwarfism, that some individuals embrace as part of their identity? And what happens when the pursuit of enhancement creates a new form of genetic inequality, forging a divide between those who can afford superior genetic endowments and those who cannot, leading to a genetically stratified society? Furthermore, we must confront the vast realm of unforeseen consequences. Our understanding of the complex interplay within the human genome, and indeed within ecosystems, is still nascent. Unintended off-target edits, unpredictable long-term health effects, and the cascading impact on human diversity and evolution are not merely theoretical concerns, but very real possibilities. We are tinkering with a system whose intricacies we do not fully comprehend, with the potential to unleash biological and societal ripple effects that could prove irreversible. This essay is an invitation to engage deeply with these critical questions. It is a call to recognize that the future of genetic engineering is not a distant, abstract problem for future generations, but an immediate and urgent challenge for ours. How we choose to wield this immense power, whether driven by caution and collective wisdom, or by unchecked ambition and market forces, will shape the very essence of humanity for centuries to come. The ethical minefields are abundant and complex, demanding not just scientific expertise, but profound moral reflection and a global, inclusive dialogue to responsibly navigate the dawn of this new age. For much of human history, our understanding of heredity was rudimentary at best, a mix of folklore and observation. Farmers and breeders, without any knowledge of genes or DNA, instinctively practiced a primitive form of genetic engineering through selective breeding. They observed that desirable traits in plants and animals, a sweeter fruit, a stronger draft horse, a more docile sheep, could be passed down through generations. By meticulously choosing which individuals to breed, they unknowingly manipulated the genetic makeup of their livestock and crops over thousands of years, transforming wild grains into staple foods and wolves into the myriad dog breeds we know today. This was genetic engineering by trial and error, a slow, often unpredictable process, yet it laid the foundational concept that traits could be managed and improved. The scientific revolution truly began in the mid-19th century with an Austrian monk named Gregor Mendel. Working tirelessly in his monastery garden, Mendel conducted meticulous experiments with pea plants. He carefully crossbred plants with distinct traits, tall or short, wrinkled or smooth peas, green or yellow pods, and painstakingly tracked how these characteristics appeared in successive generations. What Mendel discovered was revolutionary. Traits were not blended, but inherited as discrete units, which he called factors. We now know these factors as genes. Mendel's Laws of Inheritance, published in 1866, but largely overlooked for decades, provided the first scientific framework for understanding heredity, moving it from a mysterious force to a predictable biological process. The true chemical nature of Mendel's factors remained elusive until the mid-20th century. The journey to identify the molecule of heredity began in the late 19th century when Friedrich Miesser isolated nuclein from the nuclei of white blood cells. Decades later, in the 1940s, Oswald Avery, Colin MacLeod, and Maclyn McCarty conducted groundbreaking experiments demonstrating that DNA, not protein, was the substance responsible for transferring genetic information between bacteria. This was a critical turning point, though not universally accepted at first. The definitive proof came in 1952 with the Hershey-Chase experiment, which confirmed DNA as the genetic material of viruses. With DNA identified as the carrier of genetic information, the next race was to uncover its structure. Building on the crucial X-ray diffraction images captured by Rosalind Franklin and the biochemical analyses of Erwin Chargaff, James Watson and Francis Crick, in 1953, famously unveiled the double helix model of DNA. This elegant, spiraling ladder-like structure immediately explained how genetic information could be stored, replicated, and passed on with remarkable fidelity. The discovery of the double helix was not just a scientific triumph. It was the Rosetta Stone of biology, unlocking the deepest secrets of life and setting the stage for direct genetic manipulation. The understanding of DNA's structure rapidly accelerated the field of molecular biology. The 1960s saw the cracking of the genetic code, deciphering how sequences of DNA bases translate into proteins. But merely understanding the code was not enough. Scientists needed tools to cut, paste, and manipulate DNA. The breakthrough came with the discovery and isolation of restriction enzymes in the late 1960s and early 1970s by scientists like Werner Arber, Hamilton Smith, and Daniel Nathans, for which they were awarded the Nobel Prize in 1978. These enzymes act like molecular scissors, cutting DNA at very specific nucleotide sequences. This discovery was pivotal because it provided the first reliable way to cut DNA fragments at precise locations. Once DNA could be cut, the next challenge was to join pieces together. Paul Berg successfully created the first recombinant DNA molecule in 1972 by combining DNA from a monkey virus and a bacterial virus. This was the first intentional mixing and matching of DNA from different organisms. Then, in 1973, Stanley Cohen and Herbert Boyer took this a critical step further. They used restriction enzymes to cut a piece of DNA containing a gene for antibiotic resistance from one bacterium, and inserted it into a circular piece of bacterial DNA called a plasmid. They then inserted this recombinant plasmid back into another bacterium, which then acquired antibiotic resistance. This landmark experiment marked the true birth of modern genetic engineering, demonstrating the ability to transfer specific genes between organisms and have them function. The immense power of this new technology immediately raised both excitement and caution. In 1975, scientists, ethicists, and legal experts convened at the Asilomar Conference in California to discuss the potential risks and establish guidelines for responsible recombinant DNA research. This unprecedented act of scientific self-regulation was a crucial moment, setting a precedent for addressing the ethical implications of emerging biotechnologies. The 1980s saw the rapid application and commercialization of these techniques. One of the first and most impactful successes was the production of human insulin using genetically engineered bacteria in 1978 by Genentech. Before this, diabetics relied on insulin harvested from animals, which often caused allergic reactions. Genetically engineered human insulin revolutionized diabetes treatment. This decade also saw the invention of the polymerase chain reaction, or PCR, by Kary Mullis, a technique that allowed scientists to rapidly amplify tiny amounts of DNA, essentially creating millions of copies from a single strand. This unlocked new possibilities for genetic analysis, diagnostics, and forensics. As the 1990s dawned, the vision of gene therapy correcting defective genes in humans to treat disease began to materialize. In 1990, the first successful gene therapy trial was conducted on a young girl with severe combined immunodeficiency, or SCID. While early gene therapy was often challenging and sometimes faced setbacks, it demonstrated the therapeutic potential of directly targeting the genetic roots of disease. Simultaneously, genetically modified organisms, or GMOs, began to appear in agriculture, designed to be pest-resistant or herbicide-tolerant, aiming to increase crop yields and reduce reliance on chemical sprays. The culmination of much of this foundational work was the monumental Human Genome Project, launched in 1990 and completed ahead of schedule in 2003. This ambitious international collaborative effort aimed to map the entire human genome, to sequence all 3 billion base pairs of human DNA, and identify all the genes. The project provided humanity with a complete parts list of itself, an instruction manual for building a human being. This comprehensive map of our genetic landscape was not an end in itself, but rather a profound beginning, providing the crucial data and reference points necessary for the next leap in genetic engineering: precise, targeted genome editing. While early methods relied on relatively crude insertion of whole genes, the Human Genome Project paved the way for technologies that could identify and alter single letters of our genetic code with unprecedented accuracy, setting the stage for the revolutionary tools that would emerge in the 21st century. In the annals of scientific discovery, there are moments that fundamentally alter our trajectory, not just of understanding, but of capability. The advent of CRISPR-Cas9 and related gene editing technologies represents precisely such a moment for biology, and by extension, for humanity. Where previous tools for genetic manipulation were akin to clumsy surgical instruments, CRISPR burst onto the scene like a precision laser, offering unparalleled accuracy, efficiency, and accessibility in modifying DNA. This isn't merely an incremental step forward. It is a revolutionary leap that has redefined the landscape of genetic engineering, making once distant possibilities feel startlingly close. To truly grasp the transformative power of CRISPR, it's helpful to understand its remarkable origin story. Surprisingly, this sophisticated gene editing system was not engineered from scratch in a laboratory. Rather, it was discovered as a natural defense mechanism within bacteria. For billions of years, bacteria have been locked in an evolutionary arms race with viruses, known as bacteriophages, that infect and destroy them. To combat these viral invaders, bacteria evolved an ingenious adaptive immune system. When a bacterium survives a viral attack, it captures small snippets of the viral DNA and stores them within its own genome in special regions called CRISPR, arrays clustered regularly interspaced short palindromic repeats. These snippets serve as a genetic memory, like mugshots of past invaders. If that same virus, or a similar one, attempts to infect the bacterium again, the bacterium transcribes these stored viral DNA snippets into short RNA molecules. These guide RNAs then associate with a special enzyme, a protein called Cas9, CRISPR-associated protein 9. The guide RNA, with its precise match to the viral DNA, acts like a molecular GPS, navigating the Cas9 enzyme directly to the invading viral genome. Once the Cas9 guide RNA complex locates its target, the Cas9 enzyme, acting like molecular scissors, precisely cuts the viral DNA, rendering the virus harmless. This elegant bacterial defense mechanism, perfected over eons, provided the conceptual and biochemical blueprint for what has become the most powerful gene editing tool known to science. The ingenuity of scientists like Jennifer Doudna and Emmanuel Charpentier, who were awarded the Nobel Prize in Chemistry in 2020 for their work, lay in recognizing that this bacterial system could be repurposed. They realized that by designing a custom guide RNA to match any desired DNA sequence, and by pairing it with the Cas9 enzyme, they could direct these molecular scissors to any specific location within any organism's genome, be it a plant, an animal, or a human. Here's how it generally works in the lab. First, scientists synthesize a short piece of RNA, the guide RNA, that is complementary to the specific DNA sequence they want to target in the genome. This guide RNA acts as a highly specific homing beacon. Second, this guide RNA is introduced into a cell along with the Cas9 enzyme. The guide RNA then binds to the Cas9, forming a complex that scans the cell's entire genome. When the guide RNA finds its matching sequence, the Cas9 enzyme precisely unwinds the DNA double helix and makes a clean, double-stranded cut at that exact location. Once the DNA is cut, the cell's natural repair machinery springs into action, attempting to fix the break. This repair process can be leveraged in two primary ways. In one scenario, the cell tries to patch the break, but often does so imperfectly, leading to small insertions or deletions of genetic material. This effectively knocks out or inactivates the targeted gene, preventing it from producing its associated protein. This gene knockout is incredibly useful for understanding gene function or disabling a harmful gene. In the second, more sophisticated scenario, scientists can provide a new, custom-designed piece of DNA template. The cell, in its attempt to repair the break, can then use this provided template to accurately insert a new gene or correct a faulty one. This is known as knock-in or gene correction. The attributes that make CRISPR truly revolutionary are its precision, efficiency, simplicity, and versatility. Previous gene editing technologies, such as zinc finger nucleases and TALENs, offered some level of precision, but were far more complex, time-consuming, and expensive to design and implement. Imagine a finely tuned Swiss watch versus a clunky old grandfather clock. CRISPR is the watch. It is remarkably precise, minimizing the risk of off-target, edits unintended cuts at other locations in the genome. It is incredibly efficient, capable of editing multiple genes simultaneously. Crucially, it is astonishingly simple to design and deploy, requiring relatively basic molecular biology techniques and materials. This simplicity has made it accessible to thousands of laboratories worldwide, rapidly accelerating discovery and application across a multitude of fields. The impact of CRISPR-Cas9 is already profound. In basic research, it allows scientists to quickly and accurately create disease models in cell cultures and animals, enabling a deeper understanding of human diseases like cancer, Alzheimer's, and heart disease. For therapeutic purposes, CRISPR holds immense promise for treating genetic disorders. Clinical trials are underway for diseases such as sickle cell anemia, beta thalassemia, and certain forms of blindness, where scientists are attempting to correct or disable faulty genes in patients' somatic cells, those non-reproductive cells, where changes are not inherited. Imagine a future where debilitating genetic conditions could be cured with a single, precise edit. Beyond human health, CRISPR is transforming agriculture, allowing for the creation of crops with enhanced resistance to pests, diseases, and environmental stresses like drought, or with improved nutritional value. It offers new avenues for diagnostics, with systems like Sherlock and DETectr, capable of rapid, highly sensitive detection of pathogens and genetic markers. It even opens up possibilities, albeit controversial ones, for gene drives to control pest populations like malaria-carrying mosquitoes by propagating specific genetic traits through a species. However, the very ease and power that make CRISPR so revolutionary are precisely what ignite the fiercest ethical debates. The ability to precisely alter DNA in human cells, including germline cells, those that form sperm and eggs, meaning the changes would be heritable by future generations, catapults us into uncharted ethical territory. This is where the concept of designer babies moves from speculative fiction to a tangible, imminent possibility. The accessibility of CRISPR means that the ethical guardrails must be robust, globally harmonized, and continuously reevaluated. In essence, CRISPR-Cas9 has provided humanity with the ultimate biological word processor. We can now identify specific genetic typos or sentences, delete them, correct them, or even insert entirely new passages. This unprecedented control over the fundamental code of life presents us with a responsibility of monumental proportions. It forces us to confront not just what we can do, but what we should do, setting the stage for the intense ethical minefields we must now navigate. The very notion of altering our genes to combat disease might sound like science fiction. Yet it is quickly becoming a reality, representing one of the most exciting and ethically complex frontiers in modern medicine. When we talk about gene editing for therapeutic purposes, it's crucial to understand a fundamental distinction that separates two vastly different approaches, each with its own set of scientific challenges and ethical considerations. Somatic gene editing and germline gene editing. This distinction, often the first line drawn in the ethical sand, is pivotal to the entire debate surrounding genetic engineering. Let's begin with somatic gene editing. Somatic cells are essentially all the cells in your body that are not involved in reproduction: your skin cells, muscle cells, blood cells, brain cells, liver cells, and so on. When we perform somatic gene editing, we are targeting these non-reproductive cells to correct or modify a gene that is causing a specific disease in an individual patient. The key characteristic here is that any changes made to somatic cells are not heritable. They will not be passed down to that individual's children or subsequent generations. The genetic modification is confined to the treated individual. The process of somatic gene editing typically involves introducing gene editing tools like CRISPR-Cas9 into a patient's cells. This can happen in two main ways: ex vivo or in vivo. In an ex vivo approach, cells are taken out of the patient's body, genetically modified in a laboratory, and then reintroduced. A prominent example of this is in the treatment of sickle cell disease or beta thalassemia. In these conditions, patients' bone marrow stem cells, which produce all blood cells, carry a faulty gene. Scientists can extract these stem cells, use CRISPR to correct the mutation, and then infuse the corrected cells back into the patient, allowing them to produce healthy red blood cells. Similarly, for certain cancers, T cells, a type of immune cell, can be extracted, engineered to better recognize and fight cancer cells, known as CAR T cell therapy, and then reinfused. The in vivo approach, on the other hand, involves delivering the gene editing tools directly into the patient's body to target specific cells or tissues. For example, in inherited forms of blindness, where a gene mutation affects cells in the retina, gene editing components can be delivered directly into the eye using specially engineered viruses that act as delivery vehicles. Clinical trials are also exploring in vivo editing for conditions like Duchenne muscular dystrophy, where the gene editing machinery targets muscle cells. The ethical landscape surrounding somatic gene editing is, broadly speaking, much less contentious than germline editing. The primary ethical considerations here revolve around patient safety, ensuring the accuracy of the edits, minimizing off-target effects, understanding potential long-term side effects, and ensuring equitable access to these potentially life-saving therapies. While these are significant challenges, the consensus among medical professionals and bioethicists is that somatic gene therapy, when carefully researched and regulated, is a legitimate and promising avenue for treating debilitating and fatal diseases, akin to other innovative medical treatments. It offers a cure or significant improvement for a specific patient without altering the human gene pool. Now, let's turn our attention to germline gene editing. This is where the ethical discussion becomes intensely charged. Germline gene editing involves making genetic modifications to reproductive cells, sperm or egg cells, or to a very early embryo, typically within the first few days of development. The crucial, defining characteristic of germline editing is that any genetic changes made are heritable. This means the modifications would be passed down to every cell in the resulting child's body, including their own germline cells, and thus to all their future descendants, generation after generation. The potential allure of germline editing is profound. It offers the possibility of eliminating a genetic disease from a family line forever, preventing it from being passed on to any future children. For families carrying devastating inherited conditions, this might seem like the ultimate liberation. However, the ethical minefield surrounding germline editing is vast and complex, touching upon fundamental questions about human identity, consent, and societal impact. One of the most significant ethical concerns is the irreversibility and unpredictability of the changes. Unlike somatic editing, which affects only the treated individual, germline editing alters the genetic inheritance of all future generations. We simply do not have the complete understanding of the human genome's intricate workings, nor the long-term biological and evolutionary consequences of such interventions, to confidently predict the full effects across generations. Off-target edits, unintended genetic alterations, or even subtle changes that appear benign in one generation could have unforeseen and potentially detrimental effects down the line. Once these changes are introduced into the human gene pool, they are incredibly difficult, if not impossible, to undo. Another critical ethical hurdle is the absence of consent. Future individuals, whose genetic makeup would be permanently altered, have no say in the decision. This raises questions about a person's right to an open future, and whether parents, however well-intentioned, have the right to make such fundamental, unchangeable decisions about their descendants' genetic inheritance. It's a profound violation of autonomy, as the individuals who would live with these genetic modifications cannot give their consent. The safety concerns are also paramount. Beyond off-target edits, there's the risk of mosaicism, where not all cells in the embryo are successfully edited, leading to a mix of edited and unedited cells. This could result in partial correction or even new, unexpected complications. The full developmental implications of altering an early embryo's genome are still largely unknown. Perhaps the most potent ethical flashpoint is the slippery slope argument. If germline editing is permitted for therapeutic purposes, say, to eliminate a severe, debilitating disease, where do we draw the line? What if the condition is less severe? What if it's merely a predisposition? The fear is that once the door is opened for therapeutic germline editing, it will inevitably lead to the use of the technology for non-medical purposes for human enhancement, for selecting designer babies with desired traits like intelligence, athletic ability, or physical appearance. This potential transition from preventing disease to improving humanity raises specters of eugenics, a dark chapter in human history where genetics was misused to justify discrimination and forced sterilization. Furthermore, widespread germline editing could exacerbate genetic inequality. If these technologies become available, they will likely be expensive and accessible only to the wealthy. This could create a new form of class divide, a genetic haves and have-nots, where those who can afford genetic enhancements for their children gain a perceived advantage, leading to a biologically stratified society and deepening existing social disparities. Given these profound ethical challenges, there is currently a broad international consensus in most countries and among major scientific bodies that germline gene editing for reproductive purposes is ethically impermissible and should not be undertaken at this time. Many nations have explicitly banned it. This moratorium stems from a combination of safety concerns, ethical qualms about irrevocability, lack of consent, and the slippery slope leading to enhancement and eugenics. The infamous case of He Jiankui in China, who illegally created the first gene-edited babies in 2018, served as a stark, cautionary tale, illustrating the severe global repercussions and condemnation that follow when these ethical lines are crossed without societal consensus and robust oversight. In conclusion, while somatic gene editing offers a powerful and ethically more straightforward path to treating diseases within an individual's lifetime, germline gene editing carries a weight of responsibility that extends across generations. The distinction between these two approaches is not merely technical. It is the ethical fault line that defines much of the debate over the future of genetic engineering, forcing us to decide whether we are merely healing the sick or fundamentally reshaping the human species. The phrase designer babies is one that often evokes images from dystopian science fiction, a future where human beings are mass-produced with pre-selected attributes, devoid of natural variation or genuine individuality. It's a term fraught with anxiety, touching upon our deepest fears about control, identity, and the very essence of what it means to be human. But this evocative term is no longer confined to the realm of speculative fiction. With the advent of precise gene editing technologies like CRISPR-Cas9, the concept of a designer baby has transitioned from a distant nightmare to a tangible, albeit highly controversial, possibility, forcing us to confront the ethical ramifications of intentionally shaping the next generation. To understand the genesis of this concept, we must first acknowledge the morally compelling starting point for human genetic intervention: the prevention of debilitating and fatal genetic diseases. Imagine a child born with cystic fibrosis, a relentless disease that clogs the lungs and pancreas, leading to a life of chronic illness and often premature death. Or consider Huntington's disease, a devastating neurological disorder that slowly erodes a person's physical and mental capabilities in midlife. For families carrying such genetic burdens, the promise of preventing their children from inheriting these conditions is not merely an attractive scientific advancement, but a profound hope for alleviating unimaginable suffering. For decades, even before powerful gene editing tools, medical science sought ways to mitigate the impact of genetic diseases. One notable precursor to the designer baby debate, though not involving gene editing, is preimplantation genetic diagnosis, or PGD. PGD is used in conjunction with in vitro fertilization, IVF. After eggs are fertilized in a laboratory, and before implantation into the womb, a single cell can be safely removed from a several-day-old embryo. This cell is then genetically tested for specific known mutations that cause severe inherited diseases, such as Tay-Sachs, muscular dystrophy, or BRCA gene mutations predisposing to cancer. Parents can then choose to implant only those embryos that are free of the specific genetic condition they wish to avoid. While PGD involves selection of embryos rather than editing them, it fundamentally introduced the concept of genetic screening and choice at the embryonic stage, setting a precedent for intentional genetic selection. The ethical consensus on PGD for severe monogenic diseases is generally positive, seen as a responsible application of technology to prevent profound suffering. However, the leap from selecting healthy embryos to editing embryos, and from preventing disease to selecting for desirable traits, is where the ethical landscape becomes treacherous. This transition embodies what is commonly referred to as the slippery slope argument, the contention that a seemingly benign and ethically justifiable first step in genetic engineering, like preventing a severe disease, could inevitably lead to a series of progressively more ethically problematic steps, culminating in widespread human enhancement for non-medical purposes. The logic is compelling. If we can fix a gene that causes a severe disease, why not fix a gene that causes a predisposition to a less severe condition? And if that, why not a gene associated with a desired trait, like intelligence or athletic ability? The same technological capability that allows for therapeutic intervention also grants the power for enhancement. This brings us to the crucial distinction between therapy and enhancement. This is often the first and most challenging line we attempt to draw. Genetic therapy aims to restore an individual to a state of normal health or function. It seeks to correct a genetic defect that causes disease, bringing a patient from a state of illness to a baseline of health. Examples include correcting the gene mutation responsible for sickle cell anemia, or repairing a faulty gene that causes inherited blindness. The goal is clear: alleviate suffering and cure disease. Most medical ethicists and societies broadly support the pursuit of genetic therapies for serious conditions, provided they are safe and effective. Genetic enhancement, on the other hand, aims to go beyond curing disease or restoring normal function. It seeks to improve or augment human capabilities beyond what is naturally possible or typical for the species. This could involve increasing intelligence, boosting physical strength or endurance beyond elite athlete levels, extending lifespan significantly, or even altering aesthetic traits like height, hair color, or eye color. The challenge here is multifaceted. What constitutes normal or healthy when we have the tools to redefine it? Who decides what constitutes an improvement? The desire for genetic enhancement stems from a variety of sources. For parents, it's often a deeply rooted wish to give their children the best possible start in life, to provide them with advantages in an increasingly competitive world. This desire, while understandable, can quickly morph into a demand for designer traits, driven by societal pressures, perceived benefits, and even commercial incentives. If a technology could hypothetically confer a slight advantage in cognitive ability, or make a child naturally more resilient to stress? Would parents feel compelled to choose it, not out of medical necessity, but out of a desire for their child to excel? Consider the types of non-medical traits that might become targets for genetic selection or enhancement. Cognitive abilities, enhanced memory, increased processing speed, improved analytical skills, greater creativity. Physical attributes, increased muscle mass, faster metabolism, superior hand-eye coordination, resistance to fatigue, height, athletic predisposition. Aesthetic traits, specific eye or hair color, facial symmetry, body type. Behavioral personality traits, reduced aggression, increased empathy, higher resilience to stress, greater agreeableness. The very thought of deliberately engineering these traits immediately raises profound ethical questions. If we begin to select for such characteristics, what message does this send about those who possess the natural range of human variation? Does it devalue individuals with disabilities or those who do not conform to an artificially engineered ideal? Will a perfected future society be less accepting of natural human diversity, potentially leading to new forms of discrimination or prejudice against the unenhanced? Furthermore, the pursuit of designer babies for non-medical enhancement could exacerbate existing societal inequalities. Advanced genetic technologies are likely to be expensive, at least initially. This means that access to genetic improvements would likely be restricted to the wealthy, creating a new and potentially immutable form of social stratification. A future where a genetically enhanced elite coexists with a natural or unenhanced majority raises chilling questions about class, privilege, and the very concept of equal opportunity. This isn't just about income inequality. It's about potentially creating biological inequality, a divide that could become exceptionally difficult to bridge. The genesis of designer babies, therefore, lies in the tension between the humanitarian impulse to alleviate suffering through genetic therapy and the human desire for optimization and competitive advantage. It is the point where medical intervention blurs with human aspiration, and where the potential for profound good is shadowed by the risk of creating unforeseen biological and societal hierarchies. As we venture further into this genetic frontier, understanding this critical shift from therapy to enhancement, and the deep-seated ethical concerns it engenders, becomes paramount to drawing the line responsibly. In the previous section, we explored the conceptual shift from preventing genetic disease to the controversial idea of selecting for desired traits, the so-called slippery slope that leads to the notion of designer babies. We discussed how preimplantation genetic diagnosis, or PGD, already allows for selection among naturally conceived embryos based on genetic profiles, but without actively editing. Now we must confront the alarming reality that the technical capability to move beyond mere selection, to actually edit the human embryo, is not only possible, but has, disturbingly, already been attempted. The question is no longer, can we do it? But how extensively can we do it, and what are the boundaries? The transformative power of CRISPR-Cas9, which we discussed earlier, is what truly brings the designer baby concept into the realm of immediate feasibility. while PGD involves choosing an embryo from a pool based on its natural genetic lottery. CRISPR-based embryonic gene editing means we could, in theory, take any embryo and actively modify its DNA. Consider the process. In a standard IVF procedure, eggs are fertilized by sperm in a lab dish, creating embryos. At a very early stage of development, typically when an embryo is just a few cells a zygote or a blastocyst, the CRISPR-Cas9 machinery can be introduced directly into these cells. Because these are the foundational cells from which all other cells of the future organism will develop, any genetic changes made at this stage would be incorporated into every single cell of the resulting individual, including their germline cells, meaning these changes would be passed down to their children. This is the crucial point. Embryonic gene editing results in heritable changes, impacting not just one individual, but an entire lineage. The precision and efficiency of CRISPR make this prospect frighteningly real. Scientists could, for example, theoretically target a gene associated with a specific disease, such as the gene causing cystic fibrosis, and attempt to correct the mutation. But the same mechanism, the same molecular scissors, could theoretically be directed to genes that influence complex traits like intelligence or athletic ability, or even superficial characteristics like hair or eye color, if the genetic basis for such traits were fully understood. While many traits are polygenic, influenced by multiple genes, the ability to tweak even one or two genes contributing to such traits opens a Pandora's box of possibilities. This technical capability, however, came to a chilling real-world manifestation in late 2018 with the announcement by Chinese scientist He Jiankui. He shocked the global scientific and ethical communities by claiming he had created the world's first gene-edited babies. His purported goal was to confer resistance to HIV infection by editing a gene called CCR5 in human embryos. The idea was to mimic a naturally occurring mutation in the CCR5 gene, which grants some individuals a degree of resistance to certain strains of HIV. He Jiankui's announcement was met with overwhelming condemnation from scientists, ethicists, and governments worldwide. The reasons for this global outrage were multifold and profound. First and foremost, the work involved germline editing. The modifications made to the embryos were heritable, meaning they would be passed down to any future children of the two girls, named Lulu and Nana. This directly violated the near-universal international consensus that germline editing for reproductive purposes should not be undertaken due to overwhelming safety and ethical concerns. Secondly, there was a profound lack of transparency and informed consent. The research was conducted in secret, outside of established ethical review processes. The parents involved may not have fully understood the implications or risks, and certainly, the future children could not consent to being genetically altered. This undermines the very foundation of ethical research involving human subjects. Thirdly, the necessity and safety of the intervention were highly questionable. HIV infection, while serious, can be largely prevented through other means, and there are effective treatments available. The risks associated with embryonic gene editing at the time, and still today, were significant and largely unknown, including potential off-target edits, unintended changes to other parts of the genome, and unforeseen long-term health consequences for the children. Altering a gene like CCR5, even if it confers HIV resistance, could have other, unknown effects on immunity or susceptibility to other diseases. He Jianqiu's actions were seen as reckless experimentation on human beings. Fourthly, the research completely bypassed established scientific and ethical oversight. Reputable scientific bodies had called for a moratorium on germline editing precisely because of these complex issues. He Jianqui ignored these calls, demonstrating a blatant disregard for global scientific and ethical norms. The immediate repercussions of the He Jianqui case were swift and severe. He was widely denounced, lost his academic position, and was eventually sentenced to prison in China for illegal medical practice. The incident served as a stark and terrifying wake-up call, transforming the abstract debate about designer babies into a concrete reality. It demonstrated that the technical capability exists, and that if ethical guardrails are not robust and globally enforced, rogue actors can and will push boundaries with potentially devastating consequences. While the He Jianqui case focused on disease resistance, it unequivocally showed that human embryos can be edited. The mechanism used to edit a gene for HIV resistance is the same mechanism that could, in theory, be applied to genes associated with non-medical enhancing traits. This is the crux of the designer baby dilemma. The technology does not inherently distinguish between good edits curing disease and bad edits enhancing non-medical traits, or even creating unintended harm. The distinction is entirely an ethical and societal one, demanding careful consideration of where we choose to aim these powerful tools. The He Jianqui case thus stands as a critical inflection point in the narrative of genetic engineering. It stripped away the last vestiges of abstract ethical debate and placed the reality of human germline editing squarely before us. It underscored the urgent need for international dialogue, stringent regulation, and a collective decision on where humanity draws its firmest lines in the sand, lest we inadvertently slide down the very slope we fear, moving from the noble pursuit of preventing disease to the ethically fraught ambition of actively designing our descendants. We've discussed how genetic engineering holds immense promise for treating debilitating diseases. That noble pursuit, however, exists on a spectrum, and as our capabilities advance, we inevitably confront a far more provocative frontier: human enhancement. This isn't about restoring health or curing illness. It's about pushing the boundaries of what is considered normal human function, augmenting our natural capacities, and striving for a state beyond the typical. And one of the most immediate and tangible areas where this aspiration manifests is in our physical attributes. For millennia, humans have sought to improve their physical prowess, from rigorous training regimens and specialized diets to, more recently, pharmacological aids like performance-enhancing drugs. But genetic engineering offers a fundamentally different and far more profound pathway: to rewrite the very biological instructions that govern our physical capabilities. This isn't merely about training harder or taking a pill. It's about fundamentally re-engineering the blueprint of the human body itself. So, what kind of physical enhancements are we talking about? The possibilities, though still largely theoretical in humans, are inspired by natural genetic variations observed in elite athletes or even in animal models. Imagine, for instance, enhanced muscle strength and mass. In the animal kingdom, we've already seen examples that hint at this potential. Take the mighty mice created in laboratories, or the naturally occurring, double-muscled Belgian blue cattle. These animals often carry mutations in the myostatin gene. Myostatin is a protein that acts as a natural brake on muscle growth, preventing our muscles from becoming excessively large. If this gene is inhibited or knocked out, muscle mass can dramatically increase. In humans, individuals with rare mutations that inactivate myostatin have been identified, displaying exceptional muscle bulk from infancy, seemingly without adverse health effects. Genetically engineering a human to have reduced myostatin activity could, in theory, lead to significantly greater strength and muscle mass, conferring an almost superhuman physique. Beyond brute strength, consider superior endurance and aerobic capacity. Our ability to sustain physical activity is heavily reliant on how efficiently our bodies transport and utilize oxygen. One key player in this process is erythropoietin, or EPO, a hormone that stimulates the production of red blood cells. More red blood cells mean more oxygen delivered to muscles. This is why some athletes illegally dope with synthetic EPO to gain an advantage. Genetically, we might explore modifications that optimize EPO production or enhance the efficiency of oxygen uptake and utilization at a cellular level, potentially allowing individuals to run faster, swim further, or climb higher with less fatigue than ever before. Then there's enhanced bone density and resilience to injury. Our skeletons provide structure and protection, but they are susceptible to fractures and weakening over time. Certain rare genetic mutations, such as those affecting the LRP5 gene, are known to result in unusually dense bones that are remarkably resistant to fractures. While such mutations in their extreme form can lead to other complications, a more controlled genetic intervention could theoretically engineer bones that are stronger, less prone to osteoporosis, and perhaps even heal faster, making individuals more robust and resistant to trauma. The concept extends to enhanced sensory perception and pain tolerance. Imagine a genetically enhanced individual with vision far exceeding 20/20, capable of seeing in infrared or ultraviolet spectrums, or hearing frequencies beyond the typical human range. While the genetic pathways for such radical enhancements are more complex and less understood, research into the genes governing photoreceptors, auditory structures, or olfactory receptors could one day open these doors. Similarly, some individuals are born with rare genetic mutations, e.g., in the SCN9A gene, that cause a complete inability to feel pain. While this condition is extremely dangerous in its natural form, because it removes the body's crucial warning system, a controlled, partial genetic modification could theoretically allow for increased pain tolerance, enabling athletes to push through greater discomfort, or soldiers to endure severe injuries without succumbing to shock. Finally, there's the ambitious goal of enhanced disease resistance and environmental resilience. Beyond curing specific genetic diseases, genetic enhancement could aim to boost the human immune system to an unprecedented level, making individuals virtually immune to common infections, or even resistant to novel pathogens or certain cancers. Imagine genetic modifications that would make a person naturally resistant to radiation, extreme temperatures, or even the effects of aging itself, pushing the boundaries of human longevity. The prospect of such physical enhancements, however, immediately plunges us into a complex ethical quagmire. Fairness in sports and competition. If genetic engineering could confer an undeniable physical advantage, what would become of competitive sports? We already grapple with the ethics of doping, which introduces external substances to gain an edge. Genetic enhancement would be far more insidious, as the advantage would be inherent, woven into the very fabric of an individual's biology. Would unenhanced athletes be able to compete? Would genetic modification become a mandatory rite of passage for aspiring professionals, leading to an entirely new definition of fair play? The pressure to enhance and coercion. If these technologies become available and confer significant advantages, imagine the societal pressure on prospective parents to enhance their children. Would opting for a naturally conceived child be seen as neglecting their potential, or even disadvantaging them in life? This could lead to a subtle, or even overt, form of coercion, where parents feel compelled to genetically modify their offspring to ensure their success, rather than out of genuine medical necessity. Safety and unforeseen consequences. While some genetic targets like myostatin seem relatively straightforward, the human body is an incredibly complex system of interconnected biological pathways. Altering one gene, particularly at the embryonic stage, could have cascade effects, leading to unintended and potentially harmful consequences that are only discovered years, or even generations, later. Could enhanced muscle strength lead to increased strain on joints or the cardiovascular system? Could enhanced disease resistance lead to unexpected susceptibilities to other conditions? The long-term safety profile of such interventions remains a massive unknown. Defining normal and human limits, perhaps most profoundly, genetic physical enhancement forces us to ask, what defines normal human capability? If we can continuously push these limits, does it devalue the achievements of individuals who strive within natural human boundaries? Does it change our perception of human frailty, vulnerability, and the unique beauty of natural variation? These questions about physical enhancement are just the beginning of the ethical exploration. In the next part, we will delve into even more complex and abstract forms of human enhancement, particularly those targeting cognitive abilities and aesthetic traits, further challenging our understanding of human identity and societal values. Beyond the realm of physical prowess, the prospect of human enhancement stretches into territories that are even more complex and ethically fraught. The modification of cognitive abilities and aesthetic traits. These areas touch upon the very essence of human identity, our intellectual capacity, our perceptions of beauty, and the fundamental concept of individuality. When we speak of cognitive enhancement through genetic engineering, we are referring to the intentional alteration of genes to improve mental faculties beyond the typical human range. This goes far beyond existing pharmacological interventions, often dubbed smart drugs, which aim to temporarily boost alertness or focus. Genetic cognitive enhancement seeks to embed superior intellectual capabilities into an individual's biological blueprint from conception, making them an inherent part of their being. What specific cognitive traits might be targeted? The ambition here is vast. Enhanced memory. Imagine a mind capable of recalling vast amounts of information with perfect fidelity, or processing complex data sets with unprecedented speed. This could involve optimizing genes related to neurogenesis, the growth of new brain cells, or synaptic plasticity. The brain's ability to form and strengthen connections between neurons, which is fundamental to learning and memory formation. Improved learning capacity, speeding up the acquisition of new skills or knowledge. Scientists have already conducted experiments in mice, notably the Doogie mice studies, where overexpression of a specific gene involved in NMDA receptor function led to mice with significantly enhanced learning and memory. Translating such findings to humans, however, is immensely complex due to the polygenic nature of intelligence and the sheer intricacy of the human brain. Heightened attention and focus. The ability to sustain concentration for extended periods, resisting distractions, and processing multiple streams of information simultaneously. Superior problem-solving and analytical skills. Enhancing the neural networks involved in logical reasoning, critical thinking, and innovative thought. Optimized emotional regulation. While seemingly non-cognitive, our emotional states profoundly impact cognitive performance. Genetic modifications could, theoretically, lead to individuals with heightened resilience to stress, reduced predisposition to anxiety or depression, or even a predisposition to traits like increased empathy or calmness. The ethical implications of pursuing genetic cognitive enhancement are staggering. Firstly, it raises fundamental questions about the definition of intelligence and who gets to decide what constitutes an optimal mind. Will a genetically engineered intellect lead to a new intellectual elite, rendering the natural mind comparatively obsolete? This could profoundly exacerbate existing social inequalities, creating a vast chasm between the cognitively enhanced haves and the unenhanced have-nots, leading to unprecedented forms of discrimination and class stratification. Access to education, economic opportunity, and even political power could be irrevocably linked to one's genetic cognitive endowment. Secondly, there are profound psychological and societal risks. What are the psychological burdens on a child engineered for superior intelligence? Would they face immense pressure to live up to engineered expectations? Would their sense of self be tied to their genetic design, rather than their individual journey? Furthermore, fostering a societal pursuit of singular cognitive perfection could lead to a drastic loss of human neurological diversity, potentially limiting our collective capacity for different ways of thinking, problem-solving, and perceiving the world. Creativity, intuition, and emotional intelligence might be inadvertently sacrificed in the pursuit of narrow, quantifiable cognitive metrics. The brain is an incredibly complex organ. Unintended side effects of even subtle genetic changes could be catastrophic, leading to unexpected personality changes or unforeseen vulnerabilities. Moving to aesthetic enhancement, we enter an area that, while seemingly superficial, touches deeply on societal values, body image, and the commercialization of human traits. Aesthetic enhancement through genetic engineering refers to the deliberate modification of genes to achieve specific physical appearances that are considered desirable or beautiful, beyond what is typical for an individual's genetic heritage. The potential targets here include height. Genes influencing growth plates and skeletal development could be targeted to ensure a child reaches a specific, desired height. Hair and eye color. Precisely selecting or altering genes that dictate pigmentation could allow for designer hair textures or eye colors, rather than leaving it to chance. Skin tone and features. Though more complex, the genetic basis for various facial features, skin conditions, or even aspects of metabolism influencing weight distribution could theoretically be manipulated to align with prevailing beauty standards. Resistance to aging. While a form of physical enhancement, the desire for eternally youthful appearance could drive demand for genetic modifications that delay wrinkles, graying hair, or other visible signs of aging. Societies already place immense value on aesthetic traits, evidenced by the multi-billion-dollar cosmetic surgery and beauty industries. Genetic aesthetic enhancement represents the ultimate form of cosmetic intervention, embedding desired looks from conception. The ethical concerns here are equally pressing. Commodification of children. This practice would overtly treat children as products, designed to fulfill parental desires or societal trends, rather than as individuals with their own inherent value and the right to an open future. Exacerbation of unrealistic beauty standards. If genetically engineered beauty becomes attainable for some, it could create new, unattainable ideals, putting immense pressure on unenhanced individuals, and potentially leading to widespread body dissatisfaction and psychological distress. It could fuel an endless cycle of genetic keeping up with the Joneses, where parents feel compelled to give their children the latest genetically enhanced aesthetic. Loss of individuality and diversity. The natural spectrum of human appearance, with its unique variations and subtle imperfections, is part of what makes humanity rich and diverse. Widespread aesthetic engineering could lead to a homogeneous population, where differences are ironed out in favor of a narrow, artificial ideal. Shallow motivations. Is it an appropriate use of such powerful, irreversible technology to fulfill purely superficial desires? This raises questions about the allocation of scientific resources and whether humanity is directing its greatest breakthroughs toward its most trivial aspirations. In both cognitive and aesthetic enhancement, the fundamental ethical dilemma remains: the slippery slope from therapy to enhancement. Once we permit editing for non-medical reasons, where do we stop? Who regulates the menu of available traits? The transition from preventing suffering to pursuing perceived perfection carries immense risks, challenging our notions of human dignity, equality, and the inherent value of unchosen biological identity. These enhancements, unlike disease cures, push us to define what it means to be human, and whether we are prepared to fundamentally alter that definition for future generations. One of the most immediate and profound ethical minefields we must navigate in the era of advanced genetic engineering is the chilling prospect of a new, biologically entrenched class divide, leading to unprecedented forms of genetic inequality. While societies have long grappled with disparities rooted in wealth, race, religion, or geography, the ability to engineer human genes introduces a terrifying new dimension to social stratification. The potential for a fundamental biological split between those who can afford genetic enhancements and those who cannot. The genesis of this potential inequality is straightforward: access. Developing and deploying sophisticated genetic technologies, particularly those involving human germline editing or comprehensive embryonic screening, will undoubtedly be incredibly expensive, at least initially. The research and development costs are astronomical. Clinical trials are lengthy, complex, and costly. The specialized equipment, highly trained personnel, and intricate laboratory procedures required for in vitro fertilization, IVF, coupled with advanced gene editing or extensive genomic profiling, will place these technologies far beyond the reach of the average person. Consider the current landscape of advanced medical treatments. Even today, life-saving therapies for rare diseases often cost hundreds of thousands, if not millions, of dollars. Access is often limited by insurance coverage, national healthcare systems, or personal wealth. Genetic engineering, especially for enhancement purposes, is unlikely to be deemed a medical necessity that warrants universal coverage. Instead, it will likely be positioned as a high-end, elective service, available only to the super-rich. This immediately creates a scenario where the benefits of genetic engineering, whether for preventing all but the most resistant diseases or for conferring perceived advantages, are exclusively accessible to a privileged few. Their children, engineered for enhanced cognitive abilities, superior physical attributes, or heightened disease resistance, could theoretically enter the world with built-in biological advantages. Meanwhile, children born to less affluent families would continue to rely on the genetic lottery, facing the full spectrum of natural variations, including predispositions to common illnesses or merely average capabilities. The ramifications of this genetic divide are immense and deeply unsettling. Firstly, it would exacerbate existing socioeconomic disparities to an unprecedented degree. Our current inequalities are often understood as cycles of poverty, limited access to education, and systemic barriers to opportunity. While these are challenging enough to overcome, they are, in principle, surmountable. But imagine a world where the children of the wealthy are not just better educated or better connected, but also biologically smarter, healthier, or more resilient from birth. This would create a self-perpetuating cycle of advantage that would be incredibly difficult, if not impossible, for the unenhanced to break. Social mobility, already a struggle in many societies, could become virtually non-existent, replaced by a rigid, biologically determined hierarchy. Secondly, it would fundamentally undermine the concept of meritocracy. If success is increasingly determined not by effort, talent, or education, but by one's genetic endowment, then the idea of a level playing field or earning one's place in society becomes a hollow promise. Why strive for excellence if others are born with an inherent, unearned genetic edge? This could lead to widespread demoralization and disengagement among the unenhanced majority, fostering resentment and a deep sense of injustice. Thirdly, it risks creating a new form of discrimination, a genoism, where individuals are judged, valued, or marginalized based on their genetic origins. We have seen historical precedents for discrimination based on race, class, and perceived biological inferiority, often with devastating consequences. Genetic enhancement could provide a new, scientifically sanctioned basis for such prejudice. Will jobs, educational opportunities, or even social acceptance be implicitly or explicitly denied to those who are natural or unoptimized? Science fiction, notably the film Gattaca, has powerfully explored this chilling future, where one's DNA fingerprint determines one's destiny, irrespective of personal ability or character. Fourthly, this biological stratification could erode our shared sense of humanity and solidarity. What happens to empathy and social cohesion when society is bifurcated into distinct biological classes? If one group perceives itself as genetically superior, how will they relate to those they consider biologically inferior? The common bonds that tie humanity together, shared vulnerabilities, common struggles, and the inherent dignity of every human life could be fractured. This isn't just about economic disparities. It's about altering the fundamental perception of human equality and worth. Fifthly, the drive for enhancement, fueled by the desire to avoid being left behind, could create immense societal pressure on parents. Even if morally opposed, parents might feel compelled to genetically enhance their children if everyone else is doing it, fearing that their child would be disadvantaged from birth if they opted for a natural route. This subtly coercive environment, driven by market forces and perceived necessity, could undermine true parental autonomy and the celebration of genetic diversity. It's important to distinguish this potential future from historical eugenics. Classical eugenics, as practiced in the early 20th century, involved state-sponsored, often coercive programs aimed at improving the human race through selective breeding, including forced sterilization of those deemed unfit. The new genetic inequality would arise not from state mandate, but from market forces and individual consumer choice, a form of consumer eugenics. However, while the mechanism differs, the outcome a society stratified by genetic criteria, with profound implications for human rights and dignity shares chilling parallels. The ethical challenge here is immense. How do we harness the incredible therapeutic potential of genetic engineering to alleviate suffering without simultaneously creating a future where biological advantage becomes a commodity that entrenches and amplifies existing social inequalities? This requires proactive, global dialogue and robust regulatory frameworks, not just to prevent outright abuse, but to ensure that the transformative power of genetic engineering serves all of humanity, not just a privileged few. Failing to address this ethical minefield risks consigning vast segments of humanity to a permanent biological underclass, fundamentally reshaping our societies in ways we may come to profoundly regret. One of the most profound and unsettling ethical minefields opened up by the prospect of designer babies and human enhancement is the potential erosion of two foundational principles that underpin human rights and moral philosophy: human dignity and autonomy. These are not mere abstract concepts. They are the bedrock upon which our understanding of individual worth, freedom, and the moral limits of scientific intervention are built. To disregard them in the pursuit of genetic perfection risks fundamentally altering what it means to be human in ways that may prove irreversible and deeply detrimental. Let's begin with human dignity. At its core, human dignity asserts that every human being possesses an inherent, inalienable worth simply by virtue of being human. This worth is not conditional upon their abilities, appearance, social status, or genetic makeup. It is an intrinsic value, implying that individuals should be treated as ends in themselves, never merely as means to an end. It demands respect for the individual as a unique and irreplaceable being. The concept of dignity stands in direct opposition to the idea of human life as a commodity, an object to be designed, selected, or discarded based on desired specifications. The shift towards designer babies for enhancement purposes threatens this principle by ushering in the commodification of human life. When parents can choose genetic traits for their offspring, be it for intelligence, athletic prowess, or aesthetic features, children risk becoming products of design, rather than gifts to be unconditionally accepted. They are no longer simply unique individuals who emerge from the complex interplay of genetics and environment. They become made-to-order beings, customized to parental desires or societal trends. This transforms the child from a subject with inherent value into an object whose value is tied to their manufactured attributes. What happens to the love for a child if they don't live up to the genetic blueprint, or if unforeseen complications arise from the design? Does it create conditional acceptance, where parental love is contingent upon the child meeting predetermined genetic specifications? This instrumentalization of human life reduces individuals to a collection of desirable traits, stripping away their inherent, unconditional worth. Closely linked to dignity is the principle of autonomy, which refers to an individual's capacity for self-governance, to make reasoned choices about their own life, values, and identity. Genetic enhancement, particularly germline editing at the embryonic stage, presents a severe challenge to the autonomy of the future child. Consider the child's right to an open future. This concept argues that children should be afforded the maximum possible freedom to choose their own life paths, define their own identities, and pursue their own conception of the good life. When parents make irreversible genetic alterations to an embryo for non-medical reasons to enhance intelligence, athleticism, or appearance, they are, in effect, predetermining aspects of that child's life before they have any capacity for consent. The child has no say in these fundamental decisions that shape their very being. They are born into a genetic inheritance chosen for them, not by them. This creates a significant burden of engineered expectations. A child born with enhanced intelligence might feel immense pressure to excel academically, to pursue specific careers, or to live up to the lofty ambitions encoded in their DNA. This could stifle their natural inclinations, limit their exploration of diverse interests, and undermine their psychological well-being. What if the child, genetically engineered for supreme athleticism, despises sports? What if the child designed for intellectual prowess prefers a life of quiet contemplation or artistic expression? Their autonomy, their freedom to define themselves, could be severely constrained by the genetic choices made on their behalf, choices they can never undo. This is a form of genetic determinism imposed by others, robbing individuals of the spontaneous and unchosen aspects of their identity that contribute to a rich and self-directed life. Furthermore, while we often frame this debate in terms of parental autonomy, the right of parents to make choices for their children, this perspective needs careful scrutiny. Does parental autonomy extend to fundamentally altering their child's inheritable genetic makeup for non-therapeutic reasons? This is where societal pressures and market forces can subtly undermine genuine autonomy. If genetic enhancements become widespread and confer perceived advantages in a competitive world, parents might feel immense pressure to optimize their children, not out of true desire, but out of fear that their child will be disadvantaged if they don't. This isn't a free choice. It's a coerced conformity to a new norm, driven by a desire not to be left behind. This could lead to a situation where parents must choose genetic enhancement to ensure their children have a fair chance, effectively diminishing the true autonomy of both parents and children. The discussion also inevitably brings us back to the specter of eugenics. While modern genetic enhancement typically differs from the state-mandated, coercive eugenics movements of the past, which focused on eliminating undesirable traits through forced sterilization or selective breeding, it shares a troubling conceptual overlap. Both involve the notion of improving the human species by manipulating its genetic makeup according to a subjective ideal. The difference is largely in the mechanism. Historical eugenics was typically state-imposed, while modern genetic enhancement is more likely to be consumer-driven, a market-based eugenics. However, the underlying dangers defining who is fit or superior, creating genetic hierarchies, and potentially devaluing those who do not meet these engineered standards remain deeply concerning and destructive to human dignity. Finally, consider the impact on self-acceptance and the appreciation of human variation. Part of the human experience involves grappling with our natural limitations, our imperfections, and the unique combination of traits we inherit. This struggle, this journey of self-discovery and acceptance, is fundamental to personal growth and empathy. If we can simply edit away perceived flaws, does it foster a culture of perpetual dissatisfaction with anything less than perfection? Does it diminish our capacity to accept and celebrate the vast, beautiful tapestry of human diversity, including those with disabilities or genetic conditions. It risks creating a society where the inherent worth of an individual is judged not by their character or actions, but by the genetic lottery they did not choose, or by the genetic modifications chosen for them. In conclusion, the pursuit of designer babies and human enhancement, particularly through germline editing for non-therapeutic purposes, represents a profound ethical challenge to the very foundations of human dignity and autonomy. It risks commodifying human life, predetermining the futures of individuals without their consent, subtly coercing parental choices, and reintroducing eugenic ideals in a new guise. Navigating this minefield requires us to prioritize the inherent worth and self-determination of every human being, recognizing that true progress lies not in perfecting our genes, but in fostering a society that values and respects the rich, unchosen tapestry of human life in all its diverse forms. One of the most profound and perhaps insidious ethical minefields associated with the future of genetic engineering, particularly the drive towards human enhancement, is the potential for it to undermine human diversity and acceptance. For all our societal rhetoric about valuing individuality and celebrating differences, the ability to genetically optimize future generations presents a powerful, almost irresistible temptation to conform to perceived ideals, potentially leading to a future where natural human variation is diminished and perhaps even devalued. To understand this concern, we must first appreciate the inherent value of genetic diversity. Biologically, diversity is strength. A broad gene pool within a species ensures resilience and adaptability to changing environments, novel pathogens, and unforeseen challenges. It acts as a reservoir of traits, some of which may be currently unremarkable or even seem disadvantageous, but which could prove vital for survival in a future, altered world. Monocultures, whether in agriculture or in biology, are inherently vulnerable. If humanity were to collectively narrow its genetic breadth by selecting for a limited set of desirable traits, we could inadvertently strip ourselves of the very biological flexibility that has allowed our species to thrive and adapt over millennia. We simply do not know which genes, or which combinations of genes, might become crucial in 100 or 1,000 years. The drive towards enhancement, however, inherently points towards a reduction of this diversity. If parents can choose to enhance their child's intelligence, athleticism, or aesthetic features, the tendency will be to select for traits that are socially, economically, or culturally valued at a given moment. This creates a powerful homogenizing pressure. Instead of the vast, beautiful spectrum of natural human variation, we could see a narrowing, a convergence towards a limited set of ideal genetic templates. Consider the implications of widespread selection for traits like height, specific athletic predispositions, or what is culturally considered attractive facial features. Over generations, this could lead to a less varied human appearance, diminishing the unique blend of characteristics that makes each individual physically distinct. More critically, if cognitive enhancements become available, the pressure to produce smarter children could lead to a narrowing of cognitive diversity. While a high IQ is often valued, human intelligence is multifaceted, encompassing creativity, emotional intelligence, practical wisdom, and diverse learning styles. A singular focus on one metric of intelligence could inadvertently weed out other forms of cognitive brilliance, making humanity collectively less inventive, less empathetic, or less adaptable to problems requiring nonlinear thought. This homogenization carries deep societal risks. What message does it send about natural variation? If a significant portion of the population is genetically enhanced for specific traits, will those born without such enhancements be seen as lesser, unoptimized, or even defective? The very existence of widespread genetic enhancement could subtly, yet powerfully, redefine normal, shifting the baseline of human expectation. This could lead to intense pressure on parents, even those ethically opposed to enhancement, to genetically modify their children lest they be perceived as disadvantaging them from birth. The choice to embrace natural variation could become a choice for social and economic marginalization. Perhaps one of the most poignant and ethically charged aspects of this minefield relates to disability rights and identity. For people living with disabilities, the prospect of genetic technologies capable of eradicating certain conditions raises profound concerns. While there is broad consensus on using gene editing to cure severe, debilitating, and fatal diseases, the line becomes blurred when we consider conditions that some communities see not merely as medical problems, but as integral to their identity and culture. Take, for example, the deaf community. For many within this community, deafness is not simply a medical defect to be cured, but a shared experience, a cultural identity, and the basis of a rich sign language and community. The idea of routinely genetically editing embryos to prevent deafness, while seemingly benevolent from a purely medical perspective, can be interpreted by some in the deaf community as a eugenic statement, that a deaf life is not worth living, or that their identity should be erased. This is a complex ethical tension between the desire to prevent perceived suffering and the risk of devaluing a form of human experience and diversity. The debate highlights the difference between the medical model of disability and the social model of disability. The medical model views disability as an individual deficit, a problem inherent in the person that needs to be fixed or cured. The social model, conversely, argues that disability is largely created by societal barriers and attitudes that exclude people with impairments. If society is structured to be accessible and inclusive, many disabilities become mere differences. From this perspective, genetically fixing impairments can be seen as an attempt to force conformity to a narrow definition of normal, rather than adapting society to accommodate human diversity. The ethical question is critical. Are we eliminating suffering, or are we eliminating people who might experience life differently, thereby diminishing the rich tapestry of human experience? What if a genetic predisposition to certain mental states, learning styles, or physical conditions, while challenging, also confers unique perspectives or abilities? Historically, conditions that were once seen as purely negative, like autism, are increasingly understood as neurodiversities, with both challenges and unique strengths. A drive to correct such genetic variations could inadvertently remove valuable ways of thinking and experiencing the world. In essence, the ethical minefield of undermining human diversity and acceptance forces us to confront uncomfortable questions. Do we have the right to narrow the genetic future of humanity based on contemporary ideals of perfection? What are the long-term, unforeseen consequences of creating a genetically more homogeneous population, especially in the face of unknown environmental or pathological challenges? And perhaps most fundamentally, how do we uphold the inherent dignity and value of every human life in all its natural variation, without implying that some lives are better or more valuable than others based on their genetic blueprint? Navigating this requires a deep commitment to inclusivity, a profound respect for natural variation, and a cautious approach to any technology that risks defining human worth by genetic criteria. The allure of precise genetic modification is undeniable. Imagine correcting a faulty gene, eliminating a debilitating disease from a family line forever. Yet, this incredible power, particularly when applied to the human germline or for enhancement, comes tethered to a profound and inescapable ethical minefield. The vast realm of unforeseen biological and evolutionary consequences. Our understanding of the human genome, despite monumental leaps, remains far from complete, and to intervene with such powerful tools is to tread into a wilderness of unknown unknowns. First and foremost, we must acknowledge the immense complexity of the human genome itself. While the Human Genome Project provided us with a comprehensive parts list, identifying all 3 billion base pairs of our DNA, it did not provide an instruction manual for how these parts interact. Think of it like having all the words in a language, but not understanding the grammar, syntax, or deeper meaning of complex sentences. Many genes are pleiotropic, meaning a single gene can influence multiple, seemingly unrelated traits. A gene edited to enhance one function might inadvertently disrupt another, or cause an unexpected side effect years down the line. We are still uncovering the intricate regulatory networks, the non-coding regions often dismissed as junk DNA, that actually play crucial roles in gene expression, and the subtle epigenetic modifications that control how genes are turned on and off without altering the underlying DNA sequence. To intervene in such a finely tuned, interdependent system with limited knowledge is to gamble with our biological future. Despite the remarkable precision of tools like CRISPR-Cas9, technical limitations and inherent biological risks remain significant. The most frequently cited concern is off-target edits. While CRISPR is designed to cut DNA at a specific sequence, it can occasionally make unintended cuts at other, similar-looking sequences elsewhere in the genome. These accidental edits could lead to new, harmful mutations, disrupt essential genes, or even activate oncogenes, potentially leading to cancer. Even if the off-target rate is low, when performing germline editing on an embryo that will develop into an entire human being, even a single unintended edit could have catastrophic consequences for that individual and all their descendants. Beyond off-target effects, there's the issue of mosaicism. When an early embryo is edited, not all cells may take up the gene editing machinery or undergo the desired change successfully. This can result in a mosaic organism, where some cells have the genetic modification and others do not. The proportion and distribution of these edited and unedited cells can vary widely, leading to unpredictable outcomes regarding the effectiveness of the edit, and potentially causing unforeseen developmental or physiological issues that are difficult to diagnose or treat. Furthermore, even if an edit is made on target, the biological outcome might not be what was intended. The context in which a gene functions is critical. A gene that works one way in a laboratory cell line might behave differently within the complex environment of a developing human embryo and organism. For example, modifying a gene to confer resistance to one pathogen might inadvertently increase susceptibility to another, or subtly alter immune function in ways that become detrimental later in life. We are essentially conducting an irreversible experiment on future generations, with an incomplete understanding of all variables. This leads us to the crucial consideration of long-term health consequences. Many genetic disorders manifest later in life, and the full effects of a genetic modification made at conception may not become apparent until decades later. Are there subtle, delayed effects on metabolism, neurological function, or the aging process that our current short-term studies cannot detect? What are the implications for interactions with environmental factors throughout a lifespan? We lack the longitudinal data to assess the true safety profile of such radical interventions across multiple generations. Once these changes are introduced into the human gene pool, they are incredibly difficult, if not impossible, to recall or undo. Beyond the individual, the deepest concerns lie in the evolutionary implications of widespread germline editing and human enhancement. For billions of years, evolution has been an undirected process, driven by natural selection acting on random mutations. This process, while seemingly inefficient, has resulted in a remarkably robust and adaptable species. By intentionally altering our germline, we are attempting to take the reins of our own evolution, but with a knowledge base that is infinitesimally small compared to the vastness of evolutionary time. One of the most significant evolutionary risks is the narrowing of the human gene pool. If societies collectively decide to select for a limited set of desirable traits, intelligence, physical prowess, disease resistance, and eliminate what are currently perceived as undesirable genes, we risk drastically reducing human genetic diversity. Genetic diversity is the raw material for adaptation. It is what allows a species to survive unforeseen environmental shifts, new pathogens, or dramatic climate changes. A genetically homogeneous population, optimized for current conditions, could be remarkably vulnerable to future challenges, for which its artificially narrowed gene pool offers no solutions. Consider the example of the sickle cell trait. In its homozygous form, it causes sickle cell anemia, a severe disease. However, in its heterozygous form, it confers a degree of resistance to malaria. In regions where malaria is endemic, this defect actually provides a significant survival advantage. If we were to universally eliminate the sickle cell gene through germline editing, we might eradicate sickle cell disease, but we would also remove a protective mechanism, against a widespread infectious disease. This illustrates that what constitutes a defect or an enhancement can be context-dependent and change with environmental pressures. We do not know which currently undesirable traits might become advantageous in an altered future. Furthermore, we must grapple with the potential for unintended evolutionary pathways. By consciously selecting certain genes and eliminating others, we are imposing strong, artificial selective pressures on the human species. This could lead to unforeseen biological imbalances, new susceptibilities, or the coevolution of pathogens that bypass our engineered resistances. We are altering a complex biological system without a full understanding of its feedback loops and long-term consequences. This is a journey into the uncharted biological future, where every step carries the risk of profound and irreversible misdirection. The ultimate unforeseen consequence is perhaps the simplest and most terrifying: the unknown unknowns. These are the biological ramifications that we cannot even conceive of with our current scientific understanding. The human body is a mosaic of incredibly complex interactions between genes, proteins, cells, organs, and the environment. Any intervention, no matter how targeted, carries the potential for ripple effects across this intricate network that we are not yet equipped to predict. We could be opening biological doors that, once opened, cannot be closed, fundamentally reshaping the human species in ways that future generations may deem catastrophic. In summary, while the promise of genetic engineering is immense, particularly for therapeutic applications, the application of this power to the human germline or for enhancement demands extreme caution. The biological and evolutionary uncertainties are not minor considerations. They are central, existential risks. To proceed without a profound and humble acknowledgement of our limited understanding of life's deepest mechanisms is to gamble with the future of humanity itself. While the biological and evolutionary uncertainties of widespread genetic engineering present monumental challenges, the ethical minefield extends far beyond the cellular and molecular. The very fabric of human society, our psychological well-being, and the intricate dynamics of our relationships are equally vulnerable to profound and unforeseen ripple effects. We are not just altering genes. We are potentially reshaping human experience, perception, and our collective sense of self. One of the most immediate and agonizing psychological burdens would fall upon the children themselves, who are conceived through genetic enhancement. Imagine growing up knowing that fundamental aspects of your intelligence, athletic ability, or even your physical appearance were not the result of natural chance, but were meticulously chosen and engineered by your parents. How would this impact your sense of identity? Would you ever truly feel that you are your own person, or would you constantly grapple with the feeling of being a product, a manifestation of someone else's design? The fundamental question becomes, am I loved for who I am, or for what I was designed to be? This could lead to a pervasive sense of inauthenticity, a feeling that one's achievements are not truly one's own, but merely the predetermined outcome of a genetic blueprint. Furthermore, these designer children could face immense pressure to perform. If you are engineered for superior cognitive abilities, the societal and parental expectation to excel academically, to achieve groundbreaking intellectual feats, would be immense. Any perceived failure or deviation from the optimized path could lead to profound psychological distress, anxiety, and a feeling of not living up to one's genetic potential. The freedom to explore, to fail, and to define one's own version of success might be stifled by an invisible, yet powerful, genetic imperative. This pressure could manifest as perfectionism, burnout, or a deep-seated fear of inadequacy, even in individuals who are objectively thriving. Beyond the individual, the widespread adoption of genetic enhancement could dramatically alter the nature of parenthood and family dynamics. For millennia, parenthood has been characterized by unconditional love and acceptance, celebrating the unique and often unpredictable combination of traits a child inherits. The genetic lottery, with its joys and challenges, has been an intrinsic part of the human experience. With the ability to design a child, this relationship could subtly shift. The unconditional acceptance might be subtly replaced by conditional expectations. If a child doesn't exhibit the chosen traits to the desired degree, or if unforeseen side effects emerge from the genetic modifications, who bears the responsibility? Could it lead to a new form of parental blame or even regret? This shift could also create profound rifts within families. Imagine a family with both natural and genetically enhanced children. How would this dynamic affect sibling relationships? Would the unenhanced child feel inferior or resentful? Would the enhanced child feel a different kind of pressure or a sense of alienation from their natural peers and even their own siblings? This could sow seeds of division, not just in wider society, but at the very heart of the family unit. On a broader societal level, the pursuit of enhancement risks fundamental shifts in our cultural norms and values. We've already discussed the potential for a new class divide based on genetic privilege. But beyond economics, consider the psychological impact on those who are not enhanced. If a significant portion of society is deemed optimized, will those with natural variations, average intelligence, typical athletic ability, common physical appearances be seen as somehow inferior or obsolescent? This could lead to new forms of social stigma, marginalization, and prejudice, where the unenhanced are subtly or overtly discriminated against in employment, education, and social circles. The concept of normal would be fundamentally redefined, potentially creating widespread anxiety about one's natural genetic endowment. Moreover, the very nature of achievement and effort could be undermined. If superior cognitive ability or athletic prowess can be genetically purchased or endowed from birth, how does this affect our appreciation for hard work, perseverance, and genuine talent developed through effort? Does it devalue the struggles and triumphs of those who achieve greatness through sheer dedication and natural, unenhanced ability? This could lead to a less inspiring, less relatable narrative of human success, where innate genetic advantage overshadows individual striving. There's also the risk of a cultural obsession with perfection and control. If we can fix perceived genetic flaws or improve traits, it could foster a societal mindset of dissatisfaction with anything less than optimized. This could fuel an endless cycle of genetic keeping up with the Joneses, where parents feel compelled to give their children the latest genetic upgrades just to keep pace. This could lead to a reduction in empathy for human frailty and vulnerability, and a diminished appreciation for the unique, unchosen characteristics that define each individual. Finally, consider the impact on collective human experience and shared identity. Part of what binds us as a species is our shared genetic heritage, our common vulnerabilities, and our collective journey through life, with all its inherent imperfections. If humanity bifurcates into distinct genetic classes, what happens to our sense of solidarity, our capacity for collective action, and our shared understanding of what it means to be human? This could fracture social cohesion, leading to deeper divisions and potentially even conflict between genetically divergent groups. The rich tapestry of human diversity, a source of strength and innovation throughout history, could be deliberately unraveled in pursuit of narrow, engineered ideals. In sum, the unforeseen societal and psychological ripple effects of widespread genetic enhancement are as daunting as the biological ones. They threaten to transform our deepest relationships, redefine our most cherished values, and fundamentally alter the psychological landscape of human existence. Navigating this future demands not just scientific prowess, but profound wisdom, a robust commitment to human dignity and equality, and a collective willingness to engage in difficult conversations about the very essence of our humanity. The line we draw must account for these intangible, yet profoundly impactful, consequences. The unprecedented power bestowed upon us by advanced genetic engineering, particularly the ability to edit the human germline, presents not only profound scientific opportunities and ethical quandaries, but also an urgent and complex challenge for governance. As we stand at the threshold of a potential future populated by designer babies and enhanced humans, the question of where do we draw the line translates directly into how do we regulate this technology, nationally and internationally, to ensure responsible innovation and prevent irreversible harm. The need for robust regulatory frameworks is paramount. Unlike many previous scientific advancements, the implications of germline editing are not confined to a single individual or even a generation. They are inheritable, potentially altering the very biological trajectory of our species forever. Without clear ethical guidelines and legal prohibitions, the risk of misapplication, unintended consequences, and egregious ethical breaches looms large. Currently, the landscape of national regulations concerning human germline gene editing is, to put it mildly, fragmented. There is no universally agreed-upon legal or ethical standard, leading to a patchwork of approaches across different countries and jurisdictions. This diversity reflects varying cultural values, religious beliefs, and interpretations of ethical principles. Some nations have taken a remarkably firm stance, enacting explicit legal prohibitions on germline editing. Countries like Germany and France, profoundly influenced by their historical experiences with eugenics in the 20th century, have some of the most stringent laws. Germany's Embryo Protection Act, passed in 1990, broadly prohibits any alteration of human germline cells for reproductive purposes. France similarly bans any genetic modification intended to be transmitted to descendants. These legislative acts reflect a strong ethical conviction that the human germline is inviolable and should not be tampered with, driven by a deep-seated concern about the slippery slope to enhancement and the re-emergence of eugenic practices. The United Kingdom offers a somewhat different, though still cautious, model. While it has not explicitly banned germline editing research entirely, it maintains strict oversight. The Human Fertilization and Embryology Authority (HFEA) regulates all research involving human embryos. In 2016, the HFEA granted the first license to scientists at the Francis Crick Institute to use CRISPR-Cas9 to edit human embryos, but strictly for basic research purposes, specifically to understand early human development and reduce miscarriage rates. Critically, these embryos can only be studied for up to 14 days and cannot be implanted into a woman for reproductive purposes. This approach reflects a highly regulated, incremental progression, allowing for fundamental scientific inquiry while maintaining a firm barrier against clinical application of heritable edits. In contrast, other nations have less clear-cut or even more permissive stances. Some countries simply lack specific legislation addressing advanced gene editing, operating under broader ethical guidelines that may be interpreted more flexibly. In China, for example, while there are guidelines and directives from ministries of health and science that discourage or prohibit human germline editing for reproductive purposes, these are often administrative rules rather than binding laws with severe criminal penalties. This regulatory ambiguity, coupled with a highly competitive scientific landscape and different ethical frameworks, created the environment in which the infamous He Jiankui germline editing experiment was able to occur. Even in countries like the United States, the regulatory framework is complex and often relies on a combination of federal funding restrictions and institutional ethical oversight. Federal funds in the U.S. cannot be used for research that involves creating or destroying human embryos, which effectively limits germline editing research using public money. However, private funding faces fewer restrictions, though it still requires Institutional Review Board (IRB) approval, which typically adheres to broader ethical norms that currently deem germline editing for reproduction as ethically unacceptable. Despite this, the lack of an outright federal ban across all types of funding leaves a potential loophole that could be exploited. The very nature of this fragmented national regulatory landscape creates significant challenges. One of the most pressing concerns is regulatory arbitrage, sometimes referred to as ethics tourism. This phenomenon occurs when researchers or clinics, facing strict regulations or outright bans in their home countries, simply move their operations to jurisdictions with more permissive laws or less rigorous oversight. The global reach of scientific knowledge and technology means that a ban in one country does not necessarily prevent the practice from occurring elsewhere. This makes effective national regulation immensely difficult without a degree of international consensus and coordination. The He Jiankui case is a stark example of such regulatory arbitrage, where a scientist knowingly circumvented prevailing global ethical guidelines and national directives. Another significant challenge is the rapid pace of scientific advancement. Legislation and regulatory bodies, by their very nature, tend to move slowly. The time it takes to draft, debate, and enact comprehensive laws often means that by the time a regulation is in place, the science has already progressed, potentially rendering the new rules obsolete or inadequate to address emerging capabilities. CRISPR-Cas9 itself emerged with surprising speed, catching many regulatory bodies off guard and forcing a rapid scramble to develop appropriate responses. This pace problem highlights the need for dynamic, adaptive regulatory mechanisms that can evolve alongside the science. The focus of these national and nascent international discussions overwhelmingly converges on germline editing, distinguishing it sharply from somatic gene editing. The ethical consensus for a moratorium or prohibition on clinical germline editing for reproduction is rooted in several critical distinctions: its heritability across generations, the inability to obtain consent from future individuals, the profound biological and evolutionary uncertainties, and the acute, slippery slope concern that it would inevitably lead to widespread enhancement and a new form of eugenics. These are the unique characteristics that elevate germline editing beyond typical medical interventions and demand an exceptionally cautious and globally coordinated approach to governance. The He Jiankui scandal served as a potent catalyst, brutally underscoring the inadequacy of fragmented national regulations and the urgent need for a more unified international response. It demonstrated that without robust global norms and mechanisms for enforcement, rogue actors can easily push boundaries with potentially catastrophic and irreversible consequences, threatening the very credibility of scientific research and sparking widespread public distrust. The incident spurred renewed calls from major scientific bodies worldwide for international moratoria and the development of shared ethical principles, signaling that, while national efforts are crucial, the future of genetic engineering ultimately demands a global approach to governance. This sets the stage for the next crucial aspect: how the international community grapples with this shared responsibility. In the ongoing discourse surrounding the future of genetic engineering, particularly the ethically charged realms of designer babies and human enhancement, legal and regulatory frameworks, as discussed in Part 1, are undoubtedly critical. However, laws and policies, no matter how meticulously crafted, ultimately derive their legitimacy and sustainability from a broader foundation: informed public discourse, widespread education, and a societal consensus on where humanity chooses to draw its ethical lines. Without these, even the most robust regulations risk becoming brittle, easily circumvented, or simply outpaced by scientific advancement. The simple truth is that the profound questions raised by genetic engineering are not solely scientific or legal. They are fundamentally ethical, philosophical, and societal. They touch upon our deepest values, our understanding of human nature, justice, equality, and the kind of future we wish to create for our descendants. Therefore, these decisions cannot, and should not, be left solely to scientists, ethicists, or policymakers in closed-door meetings. The public, who will ultimately live with the consequences of these technologies, must be actively engaged in shaping their trajectory. However, fostering such an informed public discourse is far from straightforward. Several significant challenges stand in the way. Firstly, the scientific complexity of genetic engineering is daunting. Concepts like CRISPR-Cas9, germline editing, polygenic traits, and epigenetic modifications are intricate and often difficult for non-specialists to grasp fully. This complexity can lead to a sense of disempowerment, or worse, to an over-reliance on sensationalized media portrayals that often distort the science, fueling either unrealistic hopes or exaggerated fears. The narrative often swings wildly between miracle cures and Frankenbabies, bypassing the crucial nuanced discussion in between. Secondly, the very topics altering human life, creating designer traits, are inherently emotionally charged and morally resonant. They often intersect with deeply held personal, religious, and philosophical beliefs about the sanctity of life, the natural order, and the role of human intervention. These deeply ingrained convictions, while vital to ethical consideration, can also make objective discussion challenging, sometimes leading to entrenched positions rather than open dialogue. Given these challenges, education becomes the bedrock of responsible governance. It is not merely about disseminating scientific facts. It is about cultivating scientific literacy, ethical reasoning, and critical thinking skills across the general population. This means demystifying the science, explaining the basics of DNA, genes, and gene editing technologies like CRISPR in accessible, accurate, and engaging ways, stripping away the jargon and sensationalism. Clarifying ethical distinctions, emphasizing the crucial differences between somatic and germline editing, between therapy and enhancement, and the unique ethical implications of each. Promoting critical engagement, encouraging the public to question sources, analyze arguments, and understand the nuances of the debate, rather than succumbing to fear-mongering or uncritical acceptance of technological hype. Integrating ethics into education, incorporating bioethics into school curricula at all levels, fostering early exposure to these complex moral dilemmas, and encouraging thoughtful discussion. The role of science communicators, educators, journalists, and even popular culture, e.g., responsible science fiction in bridging this gap between expert knowledge and public understanding, is absolutely vital. With an educated populace, the next critical step is to develop mechanisms for fostering a genuine societal consensus. This requires moving beyond simple opinion polls to embrace methods of deliberative democracy, where citizens are given the opportunity to learn, discuss, and weigh complex issues in a structured environment. Examples of such mechanisms include citizen juries and assemblies, small, demographically representative groups of citizens who are provided with balanced information from diverse experts, engage in structured deliberation, and then offer recommendations on policy. These have been successfully used in various countries to address complex scientific and ethical issues. Public consultations and town halls, broad forums that allow for wider public input, ensuring that diverse voices and perspectives are heard and recorded. National Bioethics Commissions, independent, multidisciplinary bodies established by governments to advise on ethical issues arising from advances in biology and medicine. Critically, many of these commissions actively solicit public input and often publish their findings, contributing to public education and national dialogue. They serve as trusted arbiters in navigating complex moral terrains. Beyond national efforts, the global nature of genetic engineering demands international dialogue and consensus building. Scientific discoveries do not respect national borders, nor do the ethical dilemmas they raise. The He Jiankui case, for instance, was a global ethical violation, not just a national one. International scientific academies, such as the U.S. National Academy of Medicine and the UK's Royal Society, have already convened global summits to discuss human gene editing, issuing joint statements and principles that urge caution, global dialogue, and a moratorium on heritable germline editing for reproduction. Bodies like the World Health Organization, WHO, and UNESCO also play crucial roles in facilitating cross-cultural discussions and developing international guidelines. The goal is not to create a single global superregulator, which is often impractical, but rather to foster shared ethical norms that can inform national policies and prevent regulatory arbitrage. Crucially, this discourse must be inclusive. It is not enough to hear from scientists, ethicists, and government officials. The conversation must actively seek out and amplify the voices of patient advocacy groups, who bring the lived experience of genetic diseases and the profound hope, and sometimes desperation, for cures. Disability rights advocates, who offer vital perspectives on valuing diversity and challenging definitions of normalcy and defect. Religious and spiritual leaders, who provide moral frameworks derived from diverse traditions regarding life, human nature, and creation. Philosophers and social scientists, who can articulate the deeper implications for human identity, society, and justice. Artists and writers, who can explore the human experience of these technologies in ways that resonate emotionally and intellectually with the wider public. The ultimate goal of this comprehensive process of public discourse, education, and consensus building is not necessarily to achieve absolute, universal agreement on every single aspect of genetic engineering. That is often an unrealistic expectation for such complex and value-laden issues. Rather, it is to arrive at a broadly shared societal understanding of acceptable boundaries, guiding principles, and a clear articulation of shared values that can then underpin and legitimate national and international policies. This consensus, however dynamic and evolving, provides the ethical bedrock for regulation. It empowers governments to enact laws with popular support, making them more effective and durable. It fosters a collective sense of ownership over the trajectory of these powerful technologies, ensuring that our future is shaped not by unchecked scientific ambition or commercial interests, but by considered societal wisdom. Only through such a proactive, sustained, and inclusive global conversation can humanity truly draw the line and navigate the profound ethical minefields of genetic engineering in a way that safeguards our shared humanity for generations to come? We have journeyed through the astounding landscape of genetic engineering, from its ancient roots in selective breeding to the dizzying precision of modern tools like CRISPR-Cas9. We have witnessed the transformation of a theoretical concept into a tangible capability, one that promises not just to treat disease, but to fundamentally reshape the biological identity of our species. The dawn of this new age presents humanity with an unparalleled power, the ability to rewrite the very code of life itself. This power, as we have explored, is a double-edged sword. On one side lies the immense, compelling promise of alleviating human suffering. Imagine a future largely free from the scourge of inherited diseases, cystic fibrosis, Huntington's, sickle cell anemia corrected at their genetic source, offering hope where once there was only despair. Somatic gene therapy, targeting non-inheritable cells, stands as a testament to this benevolent potential, a legitimate and ethically sound pursuit of healing. Yet, beyond therapy, the spectrum inexorably broadens into the ethically fraught territory of human enhancement. The shift from preventing disease to actively designing traits, to creating what are controversially dubbed designer babies, presents us with a profound moral reckoning. This isn't just about curing illness. It's about pushing beyond normal, engineering superior physical attributes, augmenting cognitive abilities, or even sculpting aesthetic features. The very essence of human nature, once considered sacrosanct, becomes amenable to deliberate modification. As we peer into this future, the ethical minefields become starkly apparent. We confronted the chilling prospect of a new class divide and genetic inequality, where access to these powerful technologies might create a biologically stratified society, exacerbating existing disparities and undermining the very notion of meritocracy. We wrestled with the erosion of human dignity and autonomy, pondering whether children designed to specification are commodified, and if their right to an open future is irrevocably constrained by the genetic choices made by others. And we grappled with the profound risk of undermining human diversity and acceptance, questioning whether a quest for genetic perfection would narrow our collective gene pool, devalue natural variation, and potentially marginalize those who do not conform to an artificially engineered ideal, with echoes of historical eugenics. Beyond these ethical dilemmas lie the vast and unpredictable realms of unforeseen consequences. Biologically, despite our scientific advancements, our understanding of the human genome remains incomplete. Off-target edits, mosaicism, and the pleiotropic effects of genes could lead to unintended biological repercussions that manifest decades later or impact future generations in ways we cannot currently predict. We risk inadvertently creating new vulnerabilities or disrupting complex biological systems whose intricacies we barely comprehend. The long-term evolutionary impacts of consciously narrowing our genetic diversity remain an immense unknown, potentially leaving future humanity ill-equipped to adapt to unforeseen environmental or pathological challenges. The societal and psychological ripple effects are equally daunting, What are the burdens on children who grow up knowing they were designed, perhaps feeling immense pressure to live up to engineered expectations? How will family dynamics shift when children are seen not as gifts, but as projects? Could a cultural obsession with genetic perfection lead to widespread psychological distress, an intolerance for natural human imperfections, and new forms of discrimination? The very fabric of human relationships, our shared sense of belonging, and the collective narrative of what it means to be human could be profoundly altered. The imperative for robust governance, both nationally and internationally, is therefore undeniable. The fragmented patchwork of existing regulations, the dangers of ethics tourism, and the relentless pace of scientific discovery underscore the urgent need for a more unified and adaptive approach. Yet laws alone are insufficient. True legitimacy and sustainable ethical guidance must emerge from an informed public discourse, widespread education, and a genuine societal consensus. This requires transparent communication, critical engagement with complex issues, and inclusive dialogue that integrates diverse perspectives from scientists, ethicists, patients, disability advocates, and the broader public. So, where do we draw the line in the sand? This is the central, defining question for our generation. While the precise boundaries may be debated and refined as science progresses, the core principles that must guide our decisions remain steadfast. First, a firm, immediate, and globally enforced moratorium on heritable germline editing for reproductive purposes should be maintained and strengthened. The risks to safety, consent, future autonomy, and the human gene pool are currently too great, and the consequences irreversible. This line is crucial to prevent both reckless experimentation and the slippery slope to enhancement. Second, the primary focus of genetic engineering should remain resolutely on therapeutic applications for severe, debilitating, and fatal diseases, targeting somatic cells, where interventions are confined to the individual and do not alter the human germline. This is where the profound hope of gene therapy truly lies. Third, any consideration of genetic enhancement must be approached with extreme caution, humility, and foresight. We must critically examine the motivations behind such desires, the potential for exacerbating inequality, and the fundamental question of whether we wish to redefine human nature. Our guiding principle should be to alleviate suffering, not to pursue an artificial, often socially constructed, ideal of perfection. Finally, we must cultivate a global culture of responsible innovation, one that prioritizes ethical reflection as much as scientific advancement. This means fostering ongoing, inclusive public discourse, educating future generations about these profound challenges, and building international consensus on shared ethical norms. The future of genetic engineering is no longer a distant thought experiment. It is here. The power to reshape ourselves is now within our grasp. How we choose to wield this power, whether guided by wisdom, prudence, and a profound respect for human dignity and diversity, or by ambition, hubris, and market forces, will determine not just the future of medicine, but the very essence of what it means to be human. The line in the sand is waiting to be drawn, and its demarcation will be the defining ethical challenge of our age.